Good morning, everyone. Thank you for joining us on this rainy day. I am Sandy Haas, Executive Director of UWRA, and will be serving as your Zoom hostess today. In addition to our distinguished speaker, with me today are program coordinators Millard Sussman and Wendy Way. It is my pleasure to introduce a member of the Electronic Technology Committee, the Retirement Opportunities Committee, and the Committee on Financial Matters in Retirement, Millard Sussman, who is also serves on our board of directors and is serving as our program coordinator along with Wendy Way. So please join me in welcoming Millard Sussman for the purpose of an introduction of our distinguished speaker. So Millard, it's all yours.
Thank you, Sandy. Well, welcome everybody. It's a pleasure to have all of you here. I'm glad that there are so many of you signed up and ready to go on today's important topic. It's October, but it's raining outside, which is keeping us all where we've been for the past year and a half or so, inside, looking at the four walls of our house and being angry at today's topic, which is the coronavirus. Today's speaker is Tom Friedrich. He's a professor of pathobiological sciences in the School of Veterinary Medicine. He is the head of virological services at the Wisconsin National Primate Center. He's a graduate of University of Wisconsin. He has both his bachelor's degree and his PhD degree from here. And I just learned moments ago that he took the biology core curriculum back in the old days when I was teaching biology core curriculum. So I'm pleased to take credit for much of his knowledge. And I am proud to say we have a lot of distinguished graduates from the biology core curriculum, and Tom is among those distinguished scholars. His work is on RNA viruses, influenza, Zika, and the coronavirus that has us all locked up in our houses right now. And he gave us a wonderful seminar a few months ago on coronavirus. And he is going to bring us up to date. His job is to keep an eye on the coronavirus and watch it as it evolves and keep track of its new tricks. And he is going to let us know what new tricks the coronavirus has come up with over the past few months since his last seminar. So Tom, welcome. Glad to have you aboard, and I will disappear and give you the stage.
Well, thanks very much, and definitely appreciate the strong foundation of BioCore that sort of made it possible for me to get into grad school and do what I'm going to tell you about today. So, as Millard discussed, my lab has been heavily involved since the beginning of the pandemic in what we're calling genomic epidemiology. So we take specimens that have tested positive for infection with the coronavirus SARS-CoV-2, sequence the genome of the virus. These are samples that come to us from around Wisconsin. We're plugged into a nationwide and global network of investigators who are doing similar things in their own backyards. And so together we can use the genetic fingerprints of the virus to try to understand the patterns by which it spreads through space and time. Look out for worrisome signals of virus evolution, try to understand why some strains might be more successful than others, and so forth. So what I'm going to tell you about today is first a story that is based on some work that we've done in our own lab that fits in nicely with some other things that we've been hearing about. I will tell you about a short story of my interactions with the media and going viral in not a good way. And then we'll talk a little bit more about the implications of vaccines for controlling the pandemic more broadly, and I will try to gaze a little bit into my crystal ball. and say what might happen in the future. Okay, so the title sort of gives it away. We're heading to a situation of endemicity, meaning that we're not going to get rid of COVID-19 as a disease. Like, this is a virus that is likely establishing an endemic presence in human populations worldwide, and it will continue to circulate. So what does that mean? The virus will continue to evolve. Our immune responses to it will continue to change and evolve, and our ability to modulate those responses through vaccination are going to be an important tool for us to contain the damage caused by COVID disease. So what I want to talk about today are some prospects and challenges for controlling the disease. So we'll briefly revisit and sort of with a kind of new flavor, a topic that I talked about last time, which is how is it that vaccines can protect against SARS-CoV-2, the virus? How might they prevent the development of COVID-19, the disease? And then today, I want to go back to that because it's, I think, important when we hear all this news about the effectiveness of vaccines and their ability to protect, that we have to think about, well, what does protection mean? What do we want vaccines to protect against? And it turns out that there's a number of definitions. And then the way that we interpret the information we get about effectiveness depends a lot on which definition of protection we use. So I want to go through that. We'll talk about so-called breakthrough infections, which means people who get infected despite being vaccinated, and the potential for such people to transmit the virus onwards to others. We'll think about booster shots, and I'll give you some data on boosters and sort of walk you through my own thinking on that. I actually have an appointment to get a booster shot myself later today. Thoughts on the current and future patterns of SARS-CoV-2 evolution and their implications for vaccines. We'll sort of close with that, and then hopefully have some time for discussion. And as we were talking about with the other host team before the meeting started, I would be glad to take questions as we go along. So if you want to enter questions into Q&A as we go along, Miller can ask me, and I'm happy to take them at any time. I'll try to pause at some point in the middle to catch up with questions that may have collected also. All right, so let's think a little bit about what does protection mean? So what does a COVID vaccine need to do? And what it needs to do may actually be a little bit different depending on how you define protection. So as we talked about last time, the virus, SARS coronavirus 2, binds to these cellular receptors called ACE2 to get inside of cells. So here is a cartoon of the virus with its RNA genome here in this blue color. This is the viral membrane here in this light purple. And then the spike protein sticks out from the surface of that virus particle. And its job is to attach the virus like a piece of Velcro to these ACE2 receptors that stick out from cells in the respiratory tract, but also in many cells of the body. And so then there's sort of a process that has to happen by which the virus is brought close together. The virus membrane is brought close together with the cell membrane. Here's the cell. The interior of the cell is at the bottom of this picture. And so ultimately what this process leads to after binding to the receptor is fusion between the virus membrane and the cell membrane. And then the virus genetic material and replication machinery enter the cell and can take over that cell and make more viruses. That's all the virus is really trying to do. So as I mentioned, the ACE2 receptor is expressed in many tissues of the body. So cells in many places in the body, not just the respiratory tract, have the ACE2 molecule on their surfaces. And so if they are exposed to virus, they could become infected. And this helps to explain why, at least in some people and in some animals, we see systemic infections, not just restricted to the respiratory tract. And so to prevent severe disease, like the main goal that we would have from a vaccine is to prevent severe disease and death when we're talking about dealing with the pandemic. We want to keep people alive. So in order to do that, the vaccines have to induce immune responses that protect the lower respiratory tract. Why is that important? Well, virus is transmitted from the upper respiratory tract, from your nasal tissues. When you breathe, you breathe out or sneeze out viruses, but it's really infection of cells of the lower respiratory tract that is associated with severe disease. And so when the virus infection and the immune response trying to get rid of the virus further down in the lungs, in the alveoli, leads to damage of these alveolar tissues, the alveoli that are supposed to facilitate gas exchange and allow us to breathe. If they fill with fluid and cell debris, then you can't breathe anymore, and that is when severe disease occurs. So this is sort of a zoomed-in view of some alveolar tissue, and what you see here is that gas exchange is supposed to occur. You want these alveoli to be filled with mostly air. If they fill with fluid and cell debris, then you're in trouble. So if a vaccine protects, induces immunity that can sort of prevent this process from beginning or from progressing too far, you've protected the lower respiratory tract, but it turns out there's a bunch of immunological reasons why immune responses, especially ones that are induced by parenteral vaccination, by a shot in the arm as opposed to like a nasal spray or something, the vaccines that we're getting now may not be as good at protecting the upper respiratory tract. And as I mentioned last time, there is some evidence from animal models and just this, conceptual idea that virus replication that's allowed to happen in the upper respiratory tract might still allow a vaccinated person to transmit the virus to someone else, even while they are protected against severe disease. And this was a theoretical concern. And so what do these vaccines do? They're all of the vaccines that are authorized or approved for use in the United States, they encode the SARS-CoV-2 spike protein. So here is now a cartoon showing the viral membrane here on the bottom, the outer membrane of the virus. So this is like a blown up view of this cartoon here. Here is a sort of fancy diagram of what the structure of the spike protein looks like. And the job of these vaccines is to induce antibody responses. So these antibodies that are produced by your B cells, the white blood cells or lymphocytes that are stimulated by the vaccine, they should bind to the spike protein. So they're outside of cells, these antibodies. They bind to the spike protein and they physically block its access to the ACE2 receptor in a process called virus neutralization. So the whole point of these vaccines is to stimulate this type of immune response that can neutralize the virus and prevent infection. And I showed you last time some animal model data from earlier in the pandemic. This is in a non-human primate model where they vaccinated macaques with the AstraZeneca vaccine and then looked to see whether the vaccine prevented replication of SARS-CoV-2 in the lung, which it did. So here's the control animals. They have high virus loads in the lung 3 days after infection, still 5 days after infection. Very little virus in the lung in the vaccinated animals after 1 or 2 doses. So this is the lower respiratory tract. But you can see down here, if you look in the nose, then all the animals, whether they had 1 dose or 2 doses of the vaccine or no vaccine at all in these green triangles, you can see that there are high levels of virus after inoculation and challenge of these macaques with the virus. So this was some experimental evidence from preclinical vaccine testing that vaccines might protect the lower respiratory tract, but not protect the upper respiratory tract as well. So this is why we have been concerned about transmission in vaccinated people. But I mentioned this the last time, but as I said, at that time, it was kind of a theoretical concern. We didn't have much data about it. So I'm gonna go into that in just a moment. But before I do, I want to sort of walk through what we mean when we talk about protection, because when vaccine effectiveness is described in media reports of these studies, they often don't define this. So they just say that the vaccine effectiveness is 95%, or it was 95%, and now it's declined to some other percentage. But I think you always have to have in mind when you hear those types of statistics, well, effectiveness at what? What is the definition of effectiveness? And in most cases, when you're hearing reports of reduced vaccine effectiveness, we're talking about protection against any disease, against symptomatic disease. So you can think about vaccines as protecting against many different things. So mostly we think about protecting the individual who is vaccinated. So vaccines could induce immunity that protects against any infection at all, with or without symptoms. And so we might be able to tell that by vaccinating a cohort of people, having another who are unvaccinated, and then following them over time with serial COVID testing, asking, what is the rate at which the vaccinated and unvaccinated groups start to show up with test positive infections, whether they have symptoms or not. This is how the clinical trials have been done. Or once the vaccine is more widely available, people start to have symptoms and then bring themselves in for testing. You can use what's called a test negative study design to say, well, okay, if people have symptoms of COVID and they come in and get tested, I can compare the rate of test positivity in vaccinated versus unvaccinated people. People who are vaccinated may get COVID-like symptoms, but that's not caused by COVID. It could be caused by something else. And so I can ask whether the vaccine protects people in this type of study against symptomatic illness. And then again, we can ask, are these vaccines effective against severe disease, hospitalization, or death? So when people have these outcomes, we can ask, are they vaccinated or not? And we can compare the risk, the degree to which vaccination reduces the risk of these sorts of severe outcomes. And then it's also important to remember that the vaccines are a tool for protecting the population as well. So if you are less likely to become infected in the first place, or if you're less likely to transmit the virus to someone else if you are exposed or infected, then vaccination can reduce community spread of the virus also. So these are all ways that we might define protection. And I think it's important for coverage to specify when they're talking about effectiveness, which of these outcomes they're talking about. And we'll sort of unpack that as we go along. So there's been a lot of talk recently about whether or not vaccinated people who experience breakthrough infections could transmit. I showed you some preclinical data that was from May of 2020, suggesting that it might be possible. And so this is something that we actually were able to address with some of our data here in Madison. I had a very talented postdoc, Kason Riemersma, in the lab. And in our studies, we're actually interested in asking whether virus from vaccinated individuals might show evidence of natural selection. So if people get infected despite being vaccinated, could there be evidence in their virus sequences for viral evolution to escape immunity? And we were worried that that people who got infected despite being vaccinated might have lower levels of virus in their nasal cavities. And so that might be a challenge for our sequencing project. So I asked Kason to think about ways that we could compare the amounts of virus in vaccinated versus unvaccinated people initially, so that we could take steps to be sure we could still sequence virus from vaccinated individuals. So this has, we asked this question because of the sort of sequencing implications. But as we were looking at this, there was a MMWR report, so the Morbidity and Mortality Weekly Report that comes out from CDC, described a large outbreak in Barnstable County, Massachusetts, which is where Provincetown is. It's on Cape Cod. And this outbreak was associated with large gatherings. So, people going to big parties, people attending bars, all of this stuff. And about 3/4 of the people who were involved in this large outbreak were fully vaccinated at the time they became infected. And that large proportion of people who were fully vaccinated suggests that that vaccinated people must have participated in the transmission. So some of the transmission must have occurred from people who were vaccinated. And so this study was really the reason why CDC and local public health changed their masking recommendations in the middle of the summer. Since then, there have been other outbreaks involving transmission from vaccinated persons. Specifically, this outbreak involved the Delta variant. These other outbreaks involved the Delta variant. And right about the time that we were doing our analysis, CDC had shared this data with public health agencies, but hadn't quite made it public yet. So then we decided to publicize our findings early on, and that'll come back in a moment. So in order to address this sort of question of is there a difference in the amount of virus in vaccinated versus unvaccinated people, we examined almost 700 nasal swab specimens collected for testing between June 28th and July 31st of 2021. What this graph shows you here is in these lighter red colors, the proportion of viruses sequenced in Wisconsin that belonged to the Delta lineage. And so you can see that we see very little Delta in the first 6 months of 2021. So this is the week number. So the first week of 2021, second week, third week, and so forth. But then Delta very quickly took over and immediately prior to and sort of the very beginning of our study period. So this is the time here underlined in red when we collected these specimens and looked at them. We sequenced a subset of these samples to determine what virus lineage they belong to. 90% of them collected during this time that we sequenced were Delta. And then all of the samples that I'm talking about were tested for SARS-CoV-2 at a single lab at Exact Sciences here in Madison. So we're talking really about a time when Delta became the predominant and pretty much sole virus infecting people in Wisconsin. So we're looking at Delta infections primarily here. And so what these tests give you is a numerical result, which is not reported back to the clinician or the patient. You're just told positive or negative. But behind the scenes, this is a semi-quantitative assay. And it gives you a number that is proportional to the amount of viral genetic material, viral RNA that was present in this swab sample. And we call that number the cycle threshold, the threshold cycle, or CT. So for those of you who are PCR enthusiasts, that means the number of cycles of PCR that it takes for the product, for the amplified product to appear, to become visible in this reaction. So the smaller the number of cycles, that means the more RNA there was to begin with, the faster it was able to amplify. So here we're seeing low CT values on the top of the y-axis, high on the bottom, because this means high amounts of viral RNA, low amounts of viral RNA in the sample. And what you can see here is that the amounts of viral RNA that were present in samples from unvaccinated people were pretty much exactly the same as the amounts of viral RNA present in samples from people who were vaccinated but became infected during this time point, during this time that we monitored. So again, that's like late June through July of this year. And importantly, almost 70% of people who were infected despite being vaccinated had very low CT values, suggesting very high amounts of viral RNA and very high amounts of virus. So we drew this cutoff at a CT value of 25, which is about like right here. And so everything above this is a very high viral load that previous studies had associated with the presence of infectious virus. So to ask whether there was infectious virus present in samples that had this lower CT value, higher amounts of viral RNA, we partnered with Peter Halfman, a very talented scientist in Yoshikawa Oka's lab, who was able to grow the virus for us and do a so-called plaque assay. And so he took specimens from our study with this lower CT value, made dilutions of those samples, inoculated those dilutions onto a lawn of cells in culture in a dish. And then wherever a virus lands in this experiment, the virus starts infecting cells locally and spreads outwards, starting killing cells as it goes. And we make holes or plaques in this lawn. You can count the number of plaques and estimate the amount of infectious viruses that were present in the sample. And so what all this does, so we're able to compare just under 50 samples here. And you see again, the infectious titer, the amount of infectious virus present is not different in unvaccinated or vaccinated people. So altogether, this tells us that even fully vaccinated people, should they become infected with Delta variants, they frequently have high viral loads and they can shed infectious virus. So all of that is consistent with the potential for vaccinated people to shed virus and transmit it to others if they get infected. Okay, so we also asked whether symptom status affected the amount of viral RNA that was present in vaccinated versus unvaccinated people. So a pretty large proportion of these individuals had responded to a symptom questionnaire when they submitted their samples for testing. And so people who had symptoms, which is the majority, this is why most people were tested, they had similar amounts of virus in their nasal secretions, whether they were unvaccinated or vaccinated. Interestingly, even asymptomatic people, so people who got tested, even though they were asymptomatic, they also had similar levels of virus in their nasal secretions, whether they were vaccinated or unvaccinated. And at least 8 individuals who are shown here were asymptomatic, got infected despite being vaccinated, and had high viral loads. So CT values below 25, which is consistent with the ability to shed infectious virus. So this means that at least some people can be vaccinated, become infected anyway, and shed infectious virus without having symptoms, at least for a time during their infection. So this has some important implications for precautions that we might take, especially for around vulnerable people who aren't vaccinated or maybe immune compromised, even if we're fully vaccinated. Now, you may say, well, maybe the vaccinated and unvaccinated people have different test-seeking behavior. Maybe if you are vaccinated and you develop symptoms, you're really worried that you might be infected, and you go in for testing sooner. And so we may catch people who are vaccinated who systematically are earlier in their infection and have higher viral loads. And so our data might be skewed in that way. But it turns out that in this population, as our public health colleague, Kat Grande, likes to say, Dane County in particular really likes being tested. So people are very conscientious about that. And so both vaccinated and unvaccinated people tended to seek testing within about 2 days of developing symptoms. And almost all of our specimens were collected within 6 days of illness onset, which is consistent with the ability to detect the virus in these people. So that suggests that our findings are not biased by differences in test-seeking behavior in these two groups. So altogether, we think this means that the individuals who have post-vaccination infection or breakthrough infections can transmit the Delta variant. So this just sort of summarizes what I walked through with you just now. Since our study appeared, we made it public relatively early because our colleagues in public health, Madison-Dane County, were wanting to have some publicly available data to point to when they changed their masking recommendations on the basis of the CDC data, which was not yet publicly available back in early to mid-August. So since our study was first put together, and it's available on the medRxiv as a preprint here. We've seen other studies show that vaccinated individuals with Delta infections can transmit the virus, maybe even without symptoms. We've seen new outbreak investigations that clearly show that this is a possibility. And so it seems clear to me that even those of us who are vaccinated need to take precautions, especially when we're interacting with people who might be particularly vulnerable. So maybe I see that there may be a couple of questions in the Q&A, so I could potentially pause here, and then we could go on. I'll tell you the story of how this went viral.
Okay, so I'll ask you these questions. One of them is, when a vaccinated person with no symptoms tests positive for COVID, could that mean that they have the virus only in the upper respiratory tract, but do not have any viral effects in other parts of the body? Or does a positive test always mean that the virus has infected lower respiratory tract and such, as well as upper? And the other question is a very similar question, so I'll read that too. For vaccinated people, is there more going on than just exposure to the virus? In other words, could two people vaccinated on the same day, exposed to the same level of virus at the same amount of, have the same amount of time to react to it? Do they react differently, where one develops symptoms and the other doesn't?
Yeah, these are great questions. And so to answer the last question first, absolutely, there are differences between individuals in responsiveness to all vaccines, not just this one. And so if you take two people who are vaccinated with the same vaccine on the same day, and then expose them to virus some defined day after at the same time, one may develop symptoms and the other may not. That's absolutely true. I'm going to come to some data, not from our lab, but in the rest of the talk, I'll summarize data from many other groups, and we'll get to this question of, like, is there virus in the upper respiratory tract only? So if a vaccinated person tests positive, does that necessarily mean that they have virus in the lower respiratory tract? The short answer to that is no, it doesn't. So the animal model data that I showed you very early suggests that you could be exposed to virus. So you inhale it, it passes through your upper respiratory tract. You may have replication in that upper respiratory tract, and that would cause you to test positive with or maybe without symptoms. But we would hope that the vaccines would stimulate a potent enough immune response that it controls virus replication in the lower respiratory tract and protects you from disease. So we'll talk about data supporting that, that idea from humans as we go forward. Okay, so let's talk a little bit about going viral in not the best way. So when you post your paper to bioRxiv or medRxiv, they provide all of these metrics nowadays that that show you how people are interacting with your paper. And so there's this thing that computes an attention score. And so you look at it for our paper, and it says that it's in the top 5% of all papers and other outputs that of its age that are tracked. And like, wow, that's great. People are really looking at and talking about our paper. But then you kind of like, you can actually use this to see what people are saying on the different social media platforms and so forth. There's like global engagement with the paper, like, wow, fantastic. But then you look at what people say on Twitter, for example, and a lot of people say things like this. So this is somebody who cites our paper in this link here in a conversation with others. And they say, do you honestly not know that the vaccinated and non-vaccinated are equal vectors for spreading the virus? And so what we hadn't anticipated with this work is that people who are vaccine opponents would use it to say that, well, if the viral loads are similar in unvaccinated people and vaccinated people who happen to get infected despite being vaccinated, well, then obviously what that means is that the vaccine isn't making a difference at all. And so that a lot of the sharing of our article on the social media turns out to be from people using it as a cudgel to oppose vaccination mandates, to oppose vaccination. And I think a lot of it is probably honest misunderstanding of what we said. So we're going to have to change our messaging, but some of it I suspect is motivated misconstruction of what we're saying. So we can actually use data to ask the question, does vaccination reduce transmission? So there have been a few studies on this recently. Just in the past week or so, there have been a couple of studies that address this question. And so there's a large scale study reported just recently, I can't remember the exact date, in the New England Journal. And so it shows that COVID-19 vaccines can prevent infection with Delta. So after dose 1 and after dose 2, so we compare here the vaccine effectiveness against any infection. So the effectiveness is lower after full vaccination. So here's against alpha, about 88% effective, whether we're talking about the Pfizer vaccine or the AstraZeneca vaccine, which are the ones that were compared here. But so the Delta point estimate, so this estimate of vaccine effectiveness is a little bit lower than for the alpha variant, but it's still pretty good, around 80%. And so the total vaccine effectiveness for the Pfizer was estimated being 88% against infection with Delta, 67% for the AstraZeneca vaccine against infection with Delta. And so preventing any infection at all, so this is from a test negative study design. So preventing infection would take people out of circulation, reduce transmission of Delta. So we're not, when people say, look at our data and say that the vaccine is having no effect, they are not considering the fact that there's a whole bunch of people who were vaccinated who aren't getting infected with Delta at all, whereas the unvaccinated remain susceptible. It may also be that people who test positive after vaccination can have very low virus loads. We didn't see that in our own data. We didn't see a shift towards lower virus loads in our own data, but there's a large study in the UK called the REACT study that sort of randomly sent self swabs to people across England. At a time when there was very high prevalence of Delta. Most infections were due to Delta. And what they found here was that, this is sort of hard to follow, but this is the viral load or CT value on the x-axis. This is like a histogram, so the number of people who tested positive with this value on the y-axis. The vaccinated people are in blue, unvaccinated in red. And what this shows you is that there's sort of an excess number of people who test like just barely positive, who are fully vaccinated, but they have high CT values or very low amounts of virus. So, I mean, maybe there's a set of infections that are just like barely perceptible, and you only detect them when you like randomly sample a large subset of the community. And so again, these are very low virus loads, and we don't think this is consistent. Like, these virus loads are so low, that's not consistent with the shedding of infectious virus. We wouldn't expect these people to effectively transmit virus to others. Okay. Similarly, vaccination allows people to control virus replication sooner than people who are unvaccinated. So this is a study from Singapore where they enrolled people once they tested positive. Some were vaccinated, some were not, vaccinated in green, unvaccinated in red. And what you can see here is if they test people every day after symptom onset, so all these people are symptomatic, you see that viral loads remain high in both vaccinated and unvaccinated individuals for maybe 6 or 7 days after infection, after which they begin to diverge. And so people have reduced viral loads if they're vaccinated, beginning maybe 7 or 8 days after infection, whereas there's like a long tail of virus shedding. So it may take, if we look for a CT value of 25, you can see that these cross this 25 value, a few days apart, right? And so unvaccinated people may shed infectious amounts of virus for longer than vaccinated people do. when vaccinated people get infected. So there are many ways that vaccination can reduce transmission, even if there are some people who are vaccinated who can transmit, if that all makes sense. And so just recently, there was another study that came out. This appeared on bioRxiv, the preprint server, just 2 days ago. And it addresses this very question. So this is, again, data from Public Health England, where they tested the contact of infected individuals and asked, does vaccination make a difference in transmission of virus from a positive case to those persons' contacts? And so they asked this question for people who are infected with alpha versus delta, and then where the index case, where the person who initially test positive was vaccinated, or where their contacts were vaccinated. So what we're really interested in here is this vaccination. If you have a, this is the risk level, this dotted line of an unvaccinated index case. And so then we're comparing the risk that a vaccinated index case transmits to their contacts relative to this unvaccinated case. And you can see that there's a time dependence of this. So the longer after vaccination the vaccinated index person gets infected, the lower the protective effect, the closer this line gets to the line of the unvaccinated risk. And you can see that with Delta infections on this right-hand side, if you look at the AstraZeneca vaccine, by the time you get to about 12 weeks after vaccination, the vaccination of the index case, so a vaccinated person, is every bit as likely to transmit virus to their contacts as an unvaccinated person is. So Delta does reduce the protective effect of vaccination, but vaccination does still reduce the likelihood that an infected person will transmit. And vaccination in the same study dramatically reduces the likelihood that a contact, that you will become infected if you have a known contact with somebody who is infected either with alpha or with delta. Okay. So altogether, these data show us that COVID-19 vaccines reduce transmission of the coronavirus. But if vaccinated people do happen to become infected, there's a period of time in which they may still transmit the virus to others. So we have some remaining questions from this. Why does vaccine breakthrough happen? So this gets at one of the questions that was asked in the Q&A. What makes one person more resistant to infection after vaccination than another person who received the same vaccine? Are there viral factors? Does the virus change? Is it a weak immune response, or is it a combination of these things? What is the role of fully vaccinated people in transmission? So if you have a population that has a lot of vaccinated people, like we do in Dane County, you know, do people who are vaccinated but become infected anyway, do they play more of a role in transmission here than you might have in a place where there's less vaccination? I don't know. These are all studies that need to be done. And we'll talk about this next. Can boosters reduce breakthrough infections and then reduce the likelihood of transmission from the vaccinated? All right. Should we take one more question and then do the last section?
Okay, so one question is there a sex difference between susceptibility? It says in almost every age group, men and boys die at a higher rate, 1.5 to 2 times the rate of women. Is there a difference in viral load between men and women?
That's a really good question, and I don't know the answer. These are things that people have wondered about in influenza also, and I think that it's been difficult to compare viral loads across studies because, you know, our study, we were lucky because we had one lab doing all the tests that we compared. So we controlled for, you know, differences in laboratory protocols that you might have if you tried to compare tests from across different centers. So larger scale comparisons are a bit more difficult. But, you know, are there differences in risk behavior, in like risk exposure due to occupation, or are there like inherent sex differences in either susceptibility to infection in the first place or levels of virus replication? I think these are really good questions, and I at least don't know a good answer to them. Okay, so let's talk about what happens to immunity after you get vaccinated. So we've heard a lot recently about waning immunity, and there have been a lot of questions about how do you disentangle the potential that the strength of your immune response diminishes with time after vaccination. How do you divide that from the possibility that now we have circulation of Delta, a virus that is less recognizable to the immune response? So there are studies coming in that help us address this question. So this is a study that was recently made public from Israel, where they have really excellent population level data. So they compared the rates of infection in symptomatic people, almost 34,000 people. These were fully vaccinated adults over this time span shown here, May and July of 2021. The median time since vaccination in this group of people is 146 days. Almost all of the virus circulating in Israel during this time was Delta. And so if they just split the cohort in half and said, okay, let's compare the rate at which symptomatic people are actually infected with SARS-CoV-2 and not something else, for people who were vaccinated less than the median time or longer ago than the median time. What's the difference in virus positivity? And you see here for every age group, having been infected or having been vaccinated more recently in the blue bars gives you significant better protection than having been vaccinated longer ago. So this suggests that there is a waning of the protective effect of vaccination with time since vaccination. And so just yesterday or 2 days ago, there's a paper that appeared in Lancet from Kaiser Permanente. So they compared rates of SARS-CoV-2 infection in 3.4 million people receiving their care from the Kaiser Health System from December through August. They were able to sequence viruses in a large subset of these individuals, so they know whether they had Delta, which is here, or some other variant, which is here. And then they compared the vaccine effectiveness. So like, what is the protection against symptomatic infection afforded by vaccination? And then here on the x-axis is time since full vaccination. And you can see there is a clear waning of the vaccine's ability to protect against symptomatic infection. For Delta, here's in purple, and then for other viruses in red. So this waning immunity seems to be there whether you're infected with Delta or something else. But, and this gets to the very first question that was asked, I think, the protection against severe disease persists. So in panel A here, we're looking at the time since full vaccination on the x-axis, the percent effectiveness protecting against symptomatic disease, testing positive for COVID when you have symptoms across different age groups. And you can see all of these age groups with the potential exception of the 12 to 15s, all of them experience waning immunity, waning protection as we go up to 5 months after vaccination, becoming infected at that time. But the protection against hospitalization as a proxy for severe disease, that remains steady through all of these age groups through the whole 5 months. So we're seeing a decrease in protection against any infection at all, but still solid protection against severe disease. Okay, so it appears that the effectiveness of COVID vaccines, and therefore probably the strength of our immunity, wanes with time, decreases with time. And so this, and because these recent studies were able to try to disentangle the circulation of Delta from the question of protection, it really does seem that the mechanism of this decreasing protection from vaccines is waning immunity, is an ebbing of the strength of our immune responses. So does that mean we should boost everybody? So let's look at vaccine boosters, which have been very much in the news recently. Israel is a great test case here. They've had a very strong vaccination campaign for all of 2021. Their vaccine is almost entirely Pfizer because they made a large purchase early on. And so the people who are over 60 who received their second dose of vaccine at least 5 months earlier became eligible for a third dose or a booster on the 30th of July. And then they made everyone over 12 eligible for boosters on the 30th of August, 2021. And so there's data from the first month of this program that was published in the New England Journal of Medicine in the middle of September. And this is the title of the paper right here. Here's a link to it. And here's a pretty compelling figure from that paper. So they say that over 1.1 million participants received their second dose before the 1st of March and received a booster. And so they compared the relative risk of infection using their national test registry in these 1.1 million people. So some of them received boosters, some didn't. They received them at different times. And so they, beginning at 12 days after this booster, see, this is like the fold reduction in risk associated with getting a booster. So for the first like 10 to 11 days, there's no difference in risk whether you got a booster or not. But starting around day 12 and then increasing in magnitude, we see a reduction in risk of infection, about an 11-fold reduction in any infection, and about a 20-fold reduction in the risk of severe illness for people who received boosters versus those who did not. And so this just kind of shows you from like the very nice but entirely in Hebrew national dashboard that Israel maintains. So this is like a Google translated graph here, which doesn't translate everything, but this is like cases per 100,000. So the third doses in this green color reduce severe illnesses relative to 2 doses or no vaccine in both older and younger individuals. I'm just showing you the older over 60 population here. So this is the rate of over 60s with severe illness per 100,000. Not vaccinated is shown here. So that's the majority of severe illnesses. We do see some severe illnesses in people who received only 2 doses of Pfizer. And then in people who received what they call the impulse in this translation, but the third dose, we see virtually no severe disease in this group. So 2 doses still provides effective protection, but a third dose is even stronger. And then if we look at just infection at all, here's some of that data. And so the addition of these third doses dramatically reduced the risk of infection. Now here I'm showing you the under 60s. So in the entire population, so this lowest line is the people with 3 doses. 2 doses still provide protection from infection. Virus is almost entirely Delta in Israel nowadays. And again, a lot of their infections, a significant proportion of their infections in this light blue color are people who are not vaccinated. So there's still protection afforded by 2 doses, but the protection afforded by 3 doses is even stronger. And I think it's important to emphasize that there's data from all 3 authorized or approved vaccines available in the United States that a so-called remote booster, so a third dose, some longer period of time after you got the first dose or 2 doses, provides significantly stronger immune responses and or better protection. So there's data from Moderna and Johnson & Johnson also suggesting that a third dose increases immunity, decreases risk of infection and severe disease. So altogether, this suggests that third doses, boosters, can be effective at reducing transmission because they take people out of the susceptible population. And so this is a slide put together by my friend and colleague, Dave O'Connor, sort of thinking through what are the implications of the availability of boosters in developed countries. So Israel is currently at about 70% vaccinated, more than 7 months after reaching 50%. So there has been a plateau in the rate at which new people become vaccinated. Our country is at about 62% vaccinated totally, about 4 months after reaching that 50% vaccination threshold. So there's a lot of people out there who are not vaccinated because they don't want to be, or haven't gotten around to it yet for a variety of reasons. We know that hospitalization and death is concentrated in the unvaccinated population. And right now, there are susceptible individuals who are adults not getting vaccinated quickly, except when they're being motivated by mandates. The kids like my children who are both under 12 remain at risk because there's still no approved vaccine for them. People who are immune compromised, like my father, who's the recipient of a kidney transplant, they remain at risk even if they have had boosters because of their immune compromise. So if we can reduce the chances that someone who receives a booster will be infected at all, then we sort of help provide a firewall that can reduce transmission to these vulnerable populations. And so this has really kind of changed my personal thinking about the importance of boosters. And so now that they are available for people who are at risk of infection, I think it's important to use this as a tool to reduce transmission. And then just to finish up, I will say that obviously what we've called global vaccine equity remains a major challenge. So to some approximation, it is true that that money, other resources, and vaccine supplies themselves that are used to provide third doses in developed countries means doses and resources that are not being used to give vaccine first doses and second doses to people in lower income countries. And there's a dashboard that the WHO and the UN Development Program and Oxford University maintain on vaccine equity. And what it shows you here as of late last night, the vast majority of the over 3 billion vaccine doses that have been given worldwide have been given in high and upper middle income countries. You can barely even see the proportion of doses given out worldwide in the lowest income countries here in this red line. So there's a major gap in vaccine coverage in rich and poor countries. And we know that it's going to cost more resources, more money, more people, more infrastructure to give vaccines on top of all the other healthcare that's being given in every country. And so, but if we think about the fact that the average per capita healthcare expenditure in the low income countries is about $41. So that's $41 spent per person on all healthcare in low income countries. If the average cost to buy and then deliver 2 vaccine doses in those countries is about $35, so that this is average across the world, then getting 70% of a rich country's population vaccinated would cost about 0.8% of their current healthcare spending. So about less than 1% increase in a rich country's healthcare spending to get them to 70% of the population vaccinated. This price tag, even though it's relatively low, would necessitate more than 50% increase in the per capita healthcare expenditures of low-income countries to reach about 70% vaccination. So the low-income countries cannot afford this on their own. Rich countries need to subsidize vaccine, both supply and fungible resources, to the poorer countries to allow them to vaccinate their populations, which is also important for protecting the global population, ourselves too. But the challenges here go beyond just supplying vaccine doses, and there's really a lot of infrastructure challenges. So, in developing countries, we're beginning to see, high proportions of vaccination in cities, for example, but very low proportion of vaccination in the countryside. So this is an ongoing problem that's not going to be solved very soon. It's something that we need to consider. The last little bit I will talk about is, will we need vaccine updates? So here are a couple of slides from my friend and colleague, Trevor Bedford at the Fred Hutch. Trevor was just recently announced as one of this year's recipients of a MacArthur Genius Fellowship. He is indeed a genius. This is sort of data from a brainchild of his, along with one other colleague, Richard Mayer, called nextstrain.org. If you have not played around there, I highly recommend you check it out. This aggregates the kind of sequence data that my lab and others have been generating for SARS-CoV-2 from worldwide sources, then tries to map it into a real-time phylogenetic tree, a sort of family tree of viruses circulating worldwide in real time, and tries to use this information to forecast patterns of virus evolution into the future. What you see here is the growth of the Delta clade, the Delta variants in this tree. So you have sort of short stubby branches on this tree, meaning a lot of very recent sequences are Delta. Not so many of the recent sequences are anything but Delta. And then in this graph down here, this shows you the proportion of viruses that are sequenced worldwide that are Delta or other variants. And you can see that Delta is very dramatically expanding in frequency worldwide. And so what Trevor said in the talk that he gave recently is that this and many other pieces of information lead him to conclude that Delta is outcompeting the other variants. It has an evolutionary advantage to the other variants, and it's on track to complete what we call a selective sweep. So in the not too distant future, all viruses will be Delta, and all viruses that evolve in the future will descend from these Delta viruses. They're just going to outcompete the other viruses for new people to infect. And Trevor went on to present a really novel and interesting analysis that he's been doing with folks in his group that basically tried to estimate what is the evolutionary rate of SARS-CoV-2 virus. And so what he did was compare the accumulation of non-synonymous or amino acid changing mutations in different genes in the viral genome over time. So this is like a fraction of a year. So this is like 0.18 of 2020 and 0.38 of 2020 and so on. So this is how time is shown on this axis going forward almost to today. And what you see is basically an increasing accumulation of these amino acid changing mutations, specifically in the S1 domain of spike, which attaches the virus to the receptor and is the target of antibodies. And this is exactly the kind of signal we would expect to see if this gene were diversifying under natural selection. And so the selection pressure that's causing this diversification is probably human immune responses and also the virus's ability to become more transmissible. So it's these sort of two things. So and I should mention that this dotted line is kind of the place where you would plot the sort of comparable parts of the influenza virus genome. So we know that we need to frequently update influenza virus vaccines because natural selection operates to change the sequence of that virus. And so if you were to sort of plot the score of the comparable genetic region of influenza on this plot, it would be on this dotted line. And so you can see that the sort of this accumulation of amino acid changing mutations in SARS-CoV-2 is much higher than what we see in influenza. And what Trevor and others interpret this to mean is that this virus, SARS-CoV-2, is likely capable of antigenic drift, much like influenza is. And right now, it's actually evolving at a faster rate than influenza viruses are. So all that suggests that we're not going to control this pandemic in the near future. There's a huge proportion of the global population that remains completely unvaccinated and vulnerable to any virus. And so that will provide the raw material for future viral evolution and adaptation to human immune responses, and maybe even to human changes in behavior. So I expect that vaccine updates will be required in the future, just as they are for influenza viruses. So to wrap up, SARS-CoV-2 is probably, I mean, what this last bit means is that SARS-CoV-2 is likely to become the most severe of the seasonal respiratory infections that return seasonally. I don't think we're going to get rid of it. And so it's just going to be something that we have to deal with on a recurring basis, like we do with influenza. And our control efforts are going to have to continue to evolve along with the virus, and including updated vaccines. We found that 2 vaccine doses remain effective against severe disease, but protection against any infection at all seems to diminish with time. There's another study out from some Australian colleagues that suggests that the half-life of this protection against any infection may be like 110 days or so from vaccination. We know that vaccinated people can have high viral loads, can transmit virus to others, and should take precautions accordingly. So-called remote boosters, remote in time from your initial vaccine series, enhance immunity and can help prevent community spread. And this may be particularly important in communities where there are partially vaccinated populations, where you have a substantial proportion of people who are infected or who are vaccinated who could contribute to transmission, but you're below a herd immunity threshold. And this might be exactly the type of situation where we would expect the emergence of new variants to be favored. So I come down on helping take some more people out of circulation in terms of virus susceptibility. But we need to balance the benefit of boosters with the need to vaccinate the unvaccinated, both in our country and around the world. And so I think it remains to be seen whether the wealthy countries are going to live up to some of the commitments they've already made to provide funding to help administer COVID-19 vaccines globally. And I will end there, and I'm happy to take any more questions that you have.
Well, I do have a couple of questions for you. They're sort of related. One of them is, what's the information about having a shot after having had a mild case of COVID? In the particular case, the question that's asked, the person had a mild case and then had a J&J shot several months later. Is there information on the effectiveness of that? And the second is, if you had a J&J shot, J&J isn't approved as a booster yet. What's the benefit of having, say, a Pfizer booster?
Sure. So there's a lot of questions in those questions. So I would expect immunologically there to be significant benefit to being vaccinated with any vaccine after having been infected. And so you could think of your infection as an immunological prime, as your first dose, and your vaccine as a boost. So that's already two exposures. And the first, if you're infected, that means that the virus has gone to all the cells that it would naturally infect. It has stimulated a wide range of immune responses that the vaccines don't necessarily stimulate. And it has stimulated those immune responses, throughout your respiratory tract by virtue of the virus having been present there. And so if you come back sometime later with the vaccine, that should help boost all of those immune responses. I think you would get a tremendous benefit from that. And so I think there's some data, but not as much, suggesting that's actually true. So then the question was, if you've received the Johnson & Johnson vaccine, what do you do? And there's a variety of things. So there will likely be approval in the not too distant future for Johnson & Johnson boosters. So if you want to go with a homologous boost as the sort of broadest recommendations currently are, then that will be on the table, I expect, in the not too distant future. The reality is I see no immunological reason why you could not mix and match vaccines. In fact, in the HIV space, there have been a lot of people specifically studying what we call a heterologous prime boost regimen, and seeing in preclinical trials a lot of benefit, and there's even clinical data on this, from mixing vaccine types. So there's no particular danger to that I could see, and there could even be benefit. I hope that answers those questions.
Great. One more question just came in. Low-income countries are more likely to receive vaccines from China, Russia, and India. Is that an effective approach to preventing infection and global spread?
That's an excellent question. I think to vaccinate the world, all vaccines must be on the table. And we know that some of the other vaccines are not as effective as the ones that are available to us in the United States. However, some of the other vaccines are effective. So I think that, and this is something that we've thought about in the HIV world for a long time also, like what is the minimum level of vaccine effectiveness you would need to make a dent in a severe global pandemic? 95% protection against the original strain of the virus is great and sort of beyond the level of what we had initially hoped for in the Pfizer and Moderna vaccines. And I think in some ways we were spoiled by that. But the Sinovac and the Sputnik vaccines are at least 50% effective, and maybe more so. There are viral vectored vaccines that are being produced by India that are going to be more effective than that. And I think all of those need to be on the table because we need the supply. So any vaccine, even if it is 50 to 70% effective, is going to protect people from the most severe outcomes, is going to reduce transmission. May not reduce it as much as you would like, but it'll still reduce it.
I think we're out of questions, Tom.
All right.
Well, you can't hear the applause from the audience, but I will applaud for them. It was a wonderful talk, full of new information, and we hope you will be back and give us an update sometime next year, because we have an ongoing situation, as you pointed out. This is a virus that's going to be with us for a while, and we need to keep an eye on it, and we need to figure out how to protect ourselves, perhaps as we have with the flu, year after year after year. So it's good to know that there are folks like you working to keep an eye on it and make sure we know all of the tricks that it's developing.
Well, it has many tricks indeed. I wish I could say that an updated talk will not be necessary, in the spring, but I'm afraid it probably will. I'd be happy to come back and talk with you again. And thanks a lot for having me. It was fun to put this together.
Many thanks.
Thanks, everybody.
Those of you who are about to leave, please use the leave button and wait for the evaluation form to pop up. We value your advice on topics that you think are interesting and that you would like us to develop programs for. So please let us know what ideas you might have. Thank you for attending today, and we'll see you at the next of these UWRA programs. Again, thanks, Tom.